In this lecture, we're going to examine a protein called clathrin. And clathrin is an intracellular protein that's concentrated near the membrane. And clathrin is a protein that is involved in the formation of an endosome. And this is part of the receptor-mediated endocytosis process that takes place in neurons. And the way this protein operates is that it forms something called a clathrin coat. And once again, this occurs on the interior of a cell with the intention of absorbing some extracellular particle. And that particle facilitation capture is carried out by this three-legged protein, as you can see here in this computer reconstruction at the bottom, reference to this research paper published in 2006. Now, clathrin is not connected directly to the membrane. It's attached through what are called adapter protein complexes. And this process is specifically involved in the reuptake of synaptic vesicles in the terminal portion of neurons, that portion that has just released a neurotransmitter through an exocytosis mechanism where the intracellular fusion of the synaptic vesicle took place and the cell is going to reabsorb those products. The picture you see on the bottom here, taken directly from this research paper, shows two schematic hypotheses of how clathrin forms, either as a hexagonal barrel or in a more spherical football shape. And these portions of the protein corresponding to the specific residue numbers identify unique functions of the different parts of the protein. So the clathrin protein, in terms of a structure, is summarized as having three quote unquote legs, and you can see that here on the picture on the right hand side. And each leg is made up of three distinct regions: a proximal, distal, and the final N terminus region, common to all proteins. Each leg does have what's called a heavy chain and a light chain, a heavy chain referring to a sequence of a larger number of residues that are quote unquote heavier than a smaller sequence of amino acids or residues that are lighter. And there are two isoforms to these chains. It's important to note that there are slight differences in that both have what's called an 85% similarity in terms of the residues. The first isoform, referred to as clathrin heavy chain 17, or simply CHC 17, is estimated to have 1,675 residues or amino acids. And this is the isoform that's involved in membrane trafficking as well as assisting with mitosis. The other isoform, CHC22, is made up of a slightly smaller number of residues. And this type of protein, this isoform, is found particularly in skeletal muscle cells, but is not involved with endocytosis. These two isoforms separately are encoded by CLTC and CLTD genes, respectively, and these ones are found on chromosome 17 and chromosome 70 or chromosome 22. The light chains, in a similar manner, there are two light chain isoforms named just simply labeled A and B, and they're associated with CHC17, not CHC22. So the light chain part A, or isoform A, is made up of 218 residues, while type B is only made up of 211 residues and have a lower degree of similarity as the heavy chains did. And in the same manner, we see that the genes that are responsible for the light chain component are located on chromosome 9 and 5, respectively, for A and B. And the formation of an endosome occurs in a very specific manner through receptor-mediated endocytosis. And so what we see here in the top left-hand corner, we have, in a very simple way, the clathrin proteins illustrated with these relatively linear or straight lines here in this drawing. And the identification of extracellular particles occurs through a specific receptor binding on the membrane of that neuron. And this general region forms what's called a coated pit. Actin and myosin proteins, which are typically found in skeletal muscle cells that are involved in muscle force, are also located in many other cells. And in this case, the interaction of actin and myosin will cause a deformation of the cell membrane and of the cell to form that uh, uh, encircling, if you will, of the extracellular particles that are to be absorbed. And once that is formed, you can see that the clathrin proteins will form that coated pit, or rather the clathrin coat here. And then as soon as the endosome is completely formed, clathrin will start to dissolve in terms of the coat and will revert back to its original shape individually. On this final slide here, what we're looking at is a presynaptic vesicle, just really to show the complexity of the many different proteins that are involved in intracellular signaling when it comes to the process of exocytosis. So in reference to motor neurons that release the neurotransmitter acetylcholine packaged up into the vesicle, there are a large portion of the proteins that are involved in that process. So this ends our lecture here on clathrin.